Well, ladies and gentlemen, Mr. Srinivasan and Professor Srinivas, it's really an enormous honor for me to be invited to give the Srinivasan Memorial Lecture. I'm told by Krish that one of the lecturers actually collapsed on stage, and so I'm going to do my very best to keep upright. And I hope I'm going to cover this difficult topic, the question of the heredity of epilepsy. And I hope I'm going to speak for about an hour, and I hope I don't bore you too much with this topic. I never met Mr. Srinivasan, but I have grown to know about him from the many dedications and articles which I have read. And I'd like to begin, therefore, first by paying tribute to Mr. TS Srinivasan and also to his extended Srinivasan family. TS Srinivasan had a career of brilliance and innovation. And as Dr. Krishnamurthy told me, his achievements were also the embodiment of compassion. His was a life devoted to advancement and improvement in industry and also in public service, and not least to neuroscience. I learned a great deal about the generosity of Mr. Srinivasan and his family in the field of neurology, and their support of a number of ventures and developments at the VHS in Chennai. I've, over the past 40 years, visited India on quite a few occasions, and in the last 15 years have been a sort of regular visitor to VHS as a guest of Professor Srinivas and also his son. And it's extraordinarily gratifying to see the flourishing state of neurological care at the hospital and the clinical work matching, as I think it now does, the best in the world. I was also very impressed on this current visit to see the newly opened Center for Advanced Rehabilitation Specialties, which was sponsored by TVS Motors, and which is an important step, I believe. I feel humbled by the qualities and example of Mr. T.S. Srinivasan and his family, but we should celebrate the achievements the family have brought to neurology through their consistent munificence and support. In this lecture, I thought I would attempt these tasks. In the first part of my talk, I'm going to give an historical overview of the major theories of heredity, starting in the year 1860, which was when modern scientific thought in neurology really began, and over the next 100 years. And in the second part of my talk, talk about the last 50 years, which were what one might call the molecular era, in which there have been phenomenal advances in the science of heredity. In the third part of my talk, I will talk about the current state, both the successes and the failures, and briefly in the last part talk about what might be future directions and the ways in which epilepsy genetic studies may develop in the future. And although in general this is going to be a celebration of the extraordinary advances in genetics, I will emphasize that many of these concepts derive from social fashion and social theory as much as from science. And furthermore, some of the theories proved erroneous or are erroneous, and in their turn had major social implications. And I'm going to emphasize both the good and the bad aspects of these social implications. The basic message is science really is never neutral, least of all genetic science. And I hope you will see that having a historical view is important, because there are lessons from history that we should not forget. Now, this is a potentially very large topic, and furthermore, in parts, it's quite technical. And what I'm going to do is try and take as broad a brush approach as possible, but it's not going to be comprehensive, it's not going to be sufficiently explained. And for these inevitable failings, I wish to apologize in advance. So let's start first with the historical overview. Now, for over 2,000 years, ever since the writing of Hippocrates, people have realized that epilepsy was an inherited disease. But it was really only around 1860 that the modern scientific method began to be used and the modern discussion of this topic was initiated. And I think it's important to remember that this was an era when there was no investigatory facilities at all. There were no scans, there was no EEG, and doctors only had the clinical history and the clinical material on which to make a diagnosis. It was indeed a time when even neurology had only just begun to exist as a specialty, and when there was no classification of neurological disease whatsoever. There was also at this time no idea how heredity worked. Although it was recognized that somehow epilepsy was inherited, how this happened was completely unknown. Darwin's Origin of the Species had been published only the year before, and Mendel, who devised his laws, devised these 10 years later. The word genetic and gene were not conceived. And yet, despite this, there were some very interesting concepts of heredity in neurological disease formed at these times, which still have resonance today. Now, the contemporary picture in relation to epilepsy can be gained from 3 books published in 1860, 1861, and 1888. And in these books, there are 4 concepts which I'd like to expand upon. The first was that the universal belief that epilepsy was always due to an interaction between inherited factors and other causes, outside factors, sometimes things like brain tumors or strokes, but sometimes environmental factors. But these factors always interacted with inherited factors. And they divided these causes into 2, what they called the predisposing causes, which was inheritance, and the exciting causes, which were these other factors. And Sievking, who always wrote in a very flowery and imaginative way, thought of it like gunpowder and a match. The gunpowder was the inheritance. The drier the gunpowder, the more likely it was to explode. And the smaller the drier it was, the smaller was needed to be the exciting factor. And this idea that there is a mixture of factors is still very important today, and we'll discuss that a bit later. Russell Reynolds considered hereditary to be the major influence in 30% of his cases, and Gowers, William Gowers, of course, who was the renowned English neurologist, thought hereditary was the overwhelming cause in over 60% of his cases. The greatest neurologist of the time, of course, was John Hewlings Jackson. And he had some very interesting ideas on causation. He accepted that there were predisposing and exciting causes. And he also accepted the predisposing cause was inherited. And his great contribution was to recognize that the inherited mechanism was hyperexcitability of the cortex. This was an amazing insight, really, at a time when there was no chemistry and there was no physiology. And he realized that the hyperexcitability was, in fact, in his view, inherited. He interestingly had no interest in other causes and thought the cause of epilepsy was the mechanism of epilepsy. And that's a very important concept, which again, I think only recently people are again focusing upon. He thought that all seizures, whatever their cause, had this final common pathway, which was overexcitability of nervous tissue, in his view, due to deranged nutrition. But this overexcitability was the cause, whatever was the more downstream influence, causes and influence. The third concept, which was very important in those days, was the concept of the neurological trait. Now, this is an interesting idea which had developed in the mid-century sometime, but by 1880, in the time that Gowers wrote, it was universally accepted. It's hardly discussed now until in recent years, until very recently, when there seem to be modern genetic concepts reinventing this idea. According to this theory, individuals didn't inherit epilepsy alone. They inherited a range of neurological conditions, of which epilepsy was only one. This inherited propensity was known as the neurological tray, and it contained various conditions, including some psychiatric disorders, sometimes other neurological disorders, some mental retardation, and also, interestingly, some personality disorders. And as Gowers wrote, it is well known that the neuropathic tendency does not always manifest itself in the same form. The chief morbid states, beside epilepsy itself, by which the same tendency is manifest, is insanity, and to a much smaller degree, other chronic forms of disease of the brain and of the spinal cord. And intemperance, too, is probably due, in many cases, to the same disposition. That's a thought, that's an idea we'll come back to later. Now, the fourth concept I want to mention, which was of great importance, and which proved to be completely erroneous, and that is the theory of degeneration. I'm going to mention it now because of the historical consequences which it had and the lessons from which we might learn from these events. Now, the theory of degeneration was originated outside medicine, more in many other types of social discourse, not least in population science. And according to the theory, there was an inherited propensity in some individuals for progressive degeneration, which became more severe as generations moved with each generation. So, for instance, and epilepsy was thought by many to be at the core of this tendency. So, for instance, one might have migraine in one generation, then in the next generation you might get epilepsy, in the next generation you may get dementia, and so on, and eventually the line would become extinct. According to these theories, degeneration brought out what were called atavistic traits, which were primitive traits which had disappeared during evolution, and which didn't manifest except in these unfortunate individuals exhibiting these traits. They didn't manifest themselves, but they were present in the germline, although not expressed in most individuals. This theory was a social theory too, as I will explain later. Now, this theory of inherited degeneration reached its peak at about the end of the 19th century with the work of Cesare Lombroso, who was not a physician but a criminologist. And his experimental method was to measure, to look at inherited characteristics, facial characteristics, bodily characteristics, and also psychological characteristics. And he made the extraordinary step in considering that criminality in 2/3 of patients was an inherited trait, and that criminals weren't made, but people didn't become criminals, but they possessed a criminal trait, and they were what he called born criminals, and that criminality was one of these atavistic throwbacks to previous stages in evolution. Now, this had come at a time when people were looking at populations too, and there was great anxiety in the European populations that with the move of the classes from the rural areas into towns, with people, the lower classes, breeding faster than the upper classes, that the population was beginning to be weakened. And in Britain, when the Boer War came and the British Army performed very badly, there was a lot of discussion that this was because of the general weakening of the population. And so Lombroso's ideas came on fertile ground. And I mention these because these are examples of these medical ideas getting into social fashion, and social fashion influencing medicine. And then in the fourth edition of his book, Lombroso made the really disastrous link between epilepsy and criminality. And he proposed that epilepsy itself was an atavistic characteristic and was a fundamental component of the criminal type. And he supported this by showing pictures of patients, people with epilepsy, and pictures of people with criminal pictures, people from prisons, and making various measurements and so on, he found that they shared similar characteristics. And indeed, he wrote that 27% of all epileptics, men, and 25% of all epileptic women had the full criminal type. Now, this is obviously completely wrong, and the theory has no basis in scientific fact. But it does, in my mind, illustrate 3 points which are interesting and valuable today. The first is that medical theories often emerge from the wider social context. As I've said, the theories of degeneration arose because of ideas of degeneration in population, and also the arts in those days were full of pictures of arts and literature, full of pictures of people with degenerative tendencies. The point was to emphasize that the scientific theories mirrored contemporary social theory, and that they were not really objective. And we like to think nowadays of science being objective, but one should recognize that that's not always the case. The second is that although they're clearly wrong and very biased, these theories were universally accepted by doctors and the profession, and indeed most scientists. And I think it demonstrates again how skeptical we should be of all scientific theory. And the final point is that the association of epilepsy with this whole business of degeneration became immensely stigmatizing. And this opened the gate for the next scientific development, which was to prove truly disastrous in relation to epilepsy. And this, of course, was the science of eugenics. Now, eugenics became the predominant hereditarian theory in the first 3 decades of the last century. Eugenics was the science of improving genetic qualities in a population by selective breeding. It developed from Darwinian theories of natural selection and survival. It was a term coined by Galton, who advocated positive eugenic solutions. In other words, not negative solutions, which I'll come on to, but positive solutions. And he wasn't interested in medicine, he was much more interested in populations. Now, medical diseases became the subject of eugenic study in about 1900. And epilepsy, because of its link to degeneration and criminality, was soon the focus and the central feature of many of these studies. And a research office was set up in Woods Hole in the United States with Charles Davenport as director. And in 1911, Davenport produced a seminal paper in which he purported to show that epilepsy was a recessive disorder inherited along with imbecility and with negative psychiatric features. Now, these are his family trees. This was his paper. There's been a lot of studies since showing that, in fact, he simplified a lot of this data, and in fact may even have fabricated some of his data. And certainly his idea that epilepsy was a recessive disorder is completely wrong. Nevertheless, it had enormous consequences, because soon after this, the idea arose, the eugenic idea arose, that if you somehow stopped people with epilepsy reproducing, then you might cut down the number of cases by selecting out the genetic disorder. Now, of course, the genetics, this is completely wrong from the point of view of modern genetics. That's not how the condition is inherited. But nevertheless, compulsory sterilization started and was soon adopted in 23 states. And I'm interested that Chris quoted Oliver Wendell Holmes, who was the Chief Justice at the time in the Supreme Court in America, because he made this statement in this extraordinary case of Bell versus Buck, where Carrie Buck was a lady with a mental retardation and epilepsy. She was forcibly sterilized. She appealed, and Wendell Holmes said it was better for all the world, instead of waiting to execute degenerate offspring for crime or let them starve for their imbecility, society should prevent them, those who are manifestly unfit from continuing their kind, and he recommended her compulsory sterilization. It turned out in the end that she wasn't even mentally handicapped, and this was a notorious case, but which opened the floodgates for enforced sterilization. Now, of course, this all ended in disaster. With the Nazi periods, eugenics became the justification for the mass murder of the mentally handicapped and the unfit, including those with epilepsy. And it was sanctioned under the guise that this was mercy killing, which would prevent the population from becoming weakened by these people. So what happened was physicians provided the names, and the victims were either gassed or poisoned. And it's been estimated that up to 700,000 hopeless cases who were living in institutions were murdered in this way. Now, when all this came to light after the war, the science of genetics in epilepsy basically became out of bounds. And for the next 20 or 30 years, very little activity occurred. This brings us really to the end of this historical period, which was in 1960, with the famous American neurologist William Lennox, who was interested in heredity, and he made one conspicuous contribution, which was the idea, reinventing really the idea, that genetics interacted with other factors to create epilepsy. And he had this very colorful concept of streams running into a river, the river being the patient with epilepsy, the streams being the various influences which lead to the condition. So some of them were genetic, some were acquired, some were environmental, and some were hereditary. And then the stream runs into a reservoir, and the height of the banks of the reservoir determine whether or not the person has a seizure, and the banks being set largely by genetics, but also by environmental causes. And this was a very colorful way of expressing what was really known about the heredity of epilepsy in about 1960. Now this brings us then to the next phase, which is the era of molecular genetics. There's a picture here of the human chromosomes, 46 of them. In fact, this number was only really fully understood to be the number in 1956. It seems an extraordinarily short time ago. And in 1953, the amazing discoveries of how genetics operated were made by Watson and Crick. They discovered that although it was known that heredity somehow inherited characteristics were somehow passed by the nucleus of a cell and probably by DNA, it wasn't realized how the whole thing was coded. And Watson and Crick devised the model of the double helix, which I guess everybody here has heard about, in which there are chains of nucleotide bases, 4 types of bases, which code for proteins. And these chains, 3 of these bases, form a codon. There are 4 different types of bases. You can have many combinations, 20 or more combinations. They code for an amino acid. The RNA copies from the DNA, which is then transfers the codon into an amino acid, and a protein is built up from a collection of amino acids. So the string of DNA and base codes are translated into strings of amino acids, which form the protein. And variations in the genetic code end up as variations in protein. And these variations sometimes cause disease. And so what you're inheriting here from this code, transferred into the protein, transferred into the body, causes the disease. Now, after this discovery was made, there was really rapid progress. By 1972, the first whole genome, the whole genetic code of an organism was published. And then the invention of DNA sequencing by Sanger was invented. In other words, a method for identifying the code. Then it was realized that there were some genes, also things called regulator genes. And by the 1970s, it was realized that they were getting close to the whole human genome. And in 2000, the first whole genome, the first whole code of a human person was published. We recognized a little bit earlier that the human and the chimpanzee differed by only 1%. And in fact, the human differs from the earthworm, really. They share 75% of genes. And one of the enormous surprises of the Genome Project, really, was how few genes there are. And there are only 20 to 12,000, 25,000 in the human genome. And so the whole genetic basis, with all the variation of human and indeed the animal kingdom, is based on these genes. And very soon we'll have the human genome for $1,000, and all of us can get our genome identified and published. So where does epilepsy fit into all this? Sorry, could you move the slide up? Maybe the... Thank you. Okay. Well, in these 50 years, there have been a number of major advances. And I think in the field of epilepsy, I've listed these here. First, all the Mendelian disorders, the genetic disorders, the single gene disorders, in which epilepsy is part of the phenotype, part of the overall clinical picture, pretty well all of them have now been identified. There are over 200, and they've all been discovered by what are called family linkage studies, where you have a family with lots of members with these conditions, and you compare the genome of the members with the condition with the genome of members without the condition, basically, and you can identify the genetic defect. Now, out of these 200 or so cases, 200 and so conditions, 13 genes were found where the persons only had epilepsy, but they didn't have any other features. And these have been called the pure epilepsies. The first one was discovered in Melbourne in 1994, and since then there have been 13 genes identified, defects of which cause pure epilepsy without all the other features of mental retardation or other conditions. And as I say, all of them have been discovered by family linkage studies. These are the 13 genes. These are the epilepsy conditions which they produce. There are two key points. The first is that these conditions are generally quite rare. And these 13 genes account for a very small number of the overall population of epilepsy, 1 in 200 people in the population have epilepsy. About 1% of those 1 in 200 has defects in one of these genes. And the second interesting feature is the same gene can produce different types of epilepsy syndromes. And that immediately tells you that it's not just a simple transcription. There must be some other complicated feature. The second fact for these are that all these genes turn out to be what are called ion channel genes. They're genes which code for a protein in the membrane of neurons. And this was an enormous shock. And for the first time, it was then postulated that epilepsy might be what's called a channelopathy, a disease of ion channels. In the meantime, enormous progress was made in the molecular basis of the membranes. And indeed, these channels, which are units which are put into the membranes, the long neuronal membranes, and which allow the passage of ions in and out of the cell. And it's the passage of these ions which changes the electrical characteristic of the cell, which allows a nerve discharge to pass along the axon. These have now been identified in 3D by crystallography, and the whole genetic basis of each of these channel genes, the whole genome structure has been identified. And defects are shown in this picture where little defects in various parts of these genes result in a change in the structure of the protein, which results in a deficiency in the passage of the ion, either too much or too little. And it's that changes the electrical characteristic of the cell. And it's the change in electrical characteristic cell which causes hyperexcitability, as Jackson put it, and which makes the cell fire off in an epileptic fashion. So here's an example where the genetics has led to an understanding of the mechanism, which has led to a plausible explanation of why a seizure occurs. And you can look at the electrical patterns of cells with defects in these genes compared with cells which are normal. And you can see their electrical pattern differs, and the way that spontaneous firing occurs in those cells also differs on the basis of these channel genes. So this was an extraordinary development. It showed that epilepsy, there were 13 genes which were found which underlay some forms of epilepsy. These were all channel genes. This affected the electrical nature of the membrane, and that you can see how that electrical disturbance immediately could translate into an epileptic seizure. And so this was a phenomenal development. Having done this, people started looking at the common epilepsies, not the ones running in families where you could do a linkage study, but they just looked at random patients with epilepsy and carried out what are called case control association studies. And this is one of the great disappointments of modern genetics in epilepsy, that these case control association studies have led absolutely nowhere. And this was a study again from Australia showing that 165 have been carried out, $100 billion spent on these, and we have 0 results. I think what it does show is that epilepsy is a complicated affair, and that except in these families, which are very exceptional, in the ordinary person with epilepsy, although there may well be a genetic basis, it's not the result of a single gene mutation. And as Steve Jones, who's a very famous biologist in London, wrote in the British Medical Journal, the mountain has laboured and brought forth a mouse. And it's very difficult to disagree with this, which is one of the major disappointments of genetics in recent years. Now, so what about the future? That's pretty well where we are now. And I'm going to end this lecture by considering a couple of examples of the complexity which exists in the genetics of epilepsy, which may help explain the paradox that although everybody agrees epilepsy must have a genetic basis, the genetic form of this basis is largely still completely undiscovered. Now, there's a couple of clues, and this is some interesting recent work which has not looked at defects in individual genes, but has looked at what are called copy number variants, which are the presence in the genetic code of duplications of genes, or sometimes deletions of bits of genes, or even whole genes. And these duplications or deletions, which sometimes occur as the thing is being copied, anybody who's ever done any massive photocopying will know that you occasionally make mistakes. Well, so does the copying of DNA. And sometimes you miss a bit, or sometimes you duplicate a bit. People have been looking at the presence of copy number variations in epilepsy, and they've been finding that some patients with epilepsy have these copy number variations. That's discovery number one. But the second most intriguing finding, really, is that it is the same copy number variants that underpin epilepsy as also underpin some of the neuropsychiatric disorders which had been in parallel studied. So, for instance, the first one found was a deletion there, which is also found in patients with schizophrenia. Then a large study was carried out finding deletions and duplications at 5 sites on the genome. And some of these sites are shared with patients who also have autism, different patients who suffer from autism. This really, to me, seems to be a reinvention of the idea that epilepsy is inherited by mechanisms by which other neurological diseases are also inherited, including some of the neuropsychiatric disorders, which is, of course, exactly what Gowers and co. were saying in the middle of the 19th century. Now, we've been for some years looking at what's called comorbidity, in which is the fact that people with epilepsy tend to have more depression and sometimes other problems associated with the epilepsy. And it has been up till now generally assumed that comorbidity was caused by having epilepsy. But now it looks as if it's possible that these associations are not causal at all, but they reflect the fact that both may have the same mechanism. And in fact, people have now been looking at whether comorbidities predate the epilepsy and therefore can't be due to the epilepsy. And in fact, it's been found that the incidence of depression, for instance, and psychosis in patients who subsequently develop epilepsies are a lot higher than it would be in a control population also. And so the so-called bidirectionality of comorbidity may be something to do with the shared genetic basis. At least that seems to be a hypothesis which would be well worth following up. Also found was using these studies of copy number variations, people have found that the expression of a gene which is affected by, say, a deletion, the expression of that gene is affected, but also a whole lot of other genes around it also seem to be affected. And there are now beginning to be recognized various ways in which this may occur. And this finding of copy number variation also reveals the potential for what might be called reverse phenotyping, where instead of looking at a person with epilepsy and trying to find the genetic basis,